Hello everyone, welcome to Decode Science. In this video, we are going to discuss about the application of the CRISPR-Cas9 technique for the genome editing. So, first of all, what is the meaning of the genome editing? So, genome editing, it is nothing but simply the insertion, deletion, uh, or any kind of a modification within the genome of a particular organism is called as the genome editing. So either we can cut a particular gene or delete a particular gene from the genome of the organism, or we can replace it with any other type of gene sequence, or we can modify the organism's genome sequence or the DNA sequence according to our requirements, and that process is called as the genome editing process. So CRISPR-Cas9 is a very useful technique for the genome editing purpose. So let's talk about it. First point is that this technique allows for the genome editing in vivo. That means we carried out this procedure within the cells, and Cas9 and guide RNA is expressed in embryonic stem cells by the experimenter to target a particular location on the genome. These embryonic cells, which can be obtained from the blastocyst, can be cultured on the Petri plate, and in order to knock out a particular gene, we can use the CRISPR-Cas9 technique. So, what we generally do, we use the CRISPR-Cas9 technique for the genome editing purpose. Okay. So, firstly, the Cas9 protein, as we have seen in our previous video, the CRISPR-Cas technique generally involve the CRISPR RNA and the Cas proteins. And in the type 2 CRISPR-Cas technique, there is the protein involved is called as the Cas9 protein. And I told you that the type 2 system is very important in the genome editing process. So here we generally use the Cas9 protein, which is a part of the type 2 CRISPR system, and a guide RNA. So as we have seen that in the type 2 system, generally CRISPR RNA is not alone, but CRISPR RNA works along with the help of the tracer RNA. So we generally what we do, we generally in the laboratory we design a CRISPR RNA with the tracer RNA, okay, and we design a RNA which includes the part of the CRISPR RNA also and the tracer RNA also, and together this RNA, this entire RNA is called as the guide RNA, as it is helpful for the Cas9 protein just to go and bind to the target DNA site and to perform its function. So it is generally expressed in the embryonic stem cells. So as we want the change, this change about this particular gene, our target gene, we want this change in the entire organism. I mean, like we want this change in each and every type of the cells of that organism. So that's why we generally perform this procedure at the embryonic stage, and we generally take out some embryonic stem cells, and with the help of the experimenter, we tag we inject this Cas9 and the guide RNA within this embryonic stem cells. So here experimenter means that is the plasmid. Okay, we design a plasmid with the help of which we can inject our Cas9 and the guide RNA within these embryonic stem cells of the target organism. So this embryonic stem cells, which is generally obtained from the blastocyst. So we all know that the blastocyst is one of the developmental stage of the organism. Now, see, these embryonic stem cells can be cultured on the Petri plate. So what we do generally, we obtain certain embryonic stem cells from the blastocyst of an organism, and we culture them on the Petri plate, and then with the help of the CRISPR-Cas9 technique, we can either knock out a particular gene, or we can even replace a particular gene within that DNA of that organism. So how we perform that? So let's see that. For this, first a complementary CRISPR RNA must be designed along with the tracer RNA. So for that, what we need to do is we firstly we design a CRISPR RNA which would be complementary to that target gene. Okay. So what will happen, for example, like if there is a organism which has a DNA sequence, for example, like this is the entire DNA sequence of a particular organism, and if this is a gene, let's call gene A. Okay, So this is the gene A, and we want to knock out this particular gene A from the genome of this organism. So what we'll do, we'll design a CRISPR RNA in our lab, which will be complementary to this gene A. Okay, very much similar to that CRISPR Cas technique, like we have seen earlier, that bacteria generally acquired a particular DNA sequence of the bacteriophage within its own DNA, and then it transcribed that DNA sequence within the into a CRISPR RNA. And just because that the CRISPR RNA shows a complementarity with that gene, so with that DNA sequence within the bacteriophage, and it go and bind to that DNA sequence in the bacteriophage. Similar to that, here we want to knock out or replace this gene A, so we'll design a CRISPR RNA which will be complementary to this gene A. But this CRISPR RNA must be designed along with the tracer RNA. So what we'll do, we'll design a CRISPR RNA which would be complemented to this this gene A, and we'll add the sequence of the tracer RNA also. So this would be the part of the CRISPR RNA, and this would be the tracer RNA, and together it is called as the guide RNA. Okay. Then what we'll do, we'll generally Then what we'll do, we generally then to introduce this required complex and desired template into the cell, a plasmid will be designed which possesses the genes for guide RNA, Cas9 protein, and desired template gene. So what will happen is, for example, as I told you, that if I want to knock out this gene, then it's totally fine. But if I want to replace this gene A by another gene B, so what I will do, I will make a template. Okay, I will make a template DNA strand, which would have the DNA sequence for that gene B, and then I will design a plasmid. That plasmid will contain the gene for the guide RNA. Okay, this guide RNA which have the complementarity with this gene A. Along with this, that plasmid will contain the gene for the Cas9 protein, and also the gene for the desired template. So, for example, like I want to insert the gene B. Okay, I want to insert the gene B instead of this gene A. So what I will do, I will design a complementary CRISPR RNA. Then I will insert this CRISPR RNA gene, not exactly the CRISPR RNA, but you can even design a DNA sequence which would have the which can transcribed into a CRISPR RNA, and then I will inject that CRISPR RNA sequence within this plasmid. Okay, within the plasmid, and then I will add the gene for the Cas9 protein. So, for example, if this is the plasmid, so first I will inject this CRISPR RNA into it. It will contain the gene for the CRISPR RNA. Okay, this is the gene for the CRISPR RNA. Then it will contain the gene for the Cas9 protein, and after that it will contain the gene for the desired template. Okay, so what will happen? Then the plasmid can be introduced into the embryonic stem cells by electroporation or viral vectors. So as we have cultured the embryonic stem cells on the Petri plate, so what we'll do, we'll inject this plasmid with the help of the electroporation. Electroporation is the process where we generally provide the electricity to the cells, because of which the permeability of the cell membrane of the cells is generally increases, and with the help of that we can inject the plasmid or any other genetic material within the cell. Or we can even use the viral vectors. The example of the viral vectors are the lentivirus and the adenovirus. So, as we'll inject this plasmid within the embryonic stem cell, what will happen? This gene for the CRISPR RNA will be get transcribed into the CRISPR RNA. Okay. Then on this Cas9 protein gene will be get transcribed and translate, and it will form the Cas9 proteins, and the desired template gene will also get replicated and all, and we have the multiple copies of the desired template. Now what will happen is that in the next stage, that the Cas9 and guide RNA associated complex is brought to the complementary sequence on the genome, and the Cas9 protein makes a double strand break. So now in the next stage, what will happen, that on the Cas9 protein and the guide RNA, okay, now the plasmid has entered into the embryonic stem cells, and the Cas9 protein has formed from the Cas9 gene sequence present within the plasmid. At the same time, the cell has guide RNAs. So this guide RNA will go and will bind to that target gene, like here. Okay, this CRISPR RNA will be get transcribed, and then it will go and it will bind to that gene A. Then on the basis of that, the Cas9 protein will recognize it and will cut the DNA at this sequence and will knock out this gene A and will create a double strand break, okay, within this DNA sequence of that target organism. After that, this DSB can be repaired by the non-homologous end joining or DSB model, that is the double strand break model, which is also known as the homology directed repair mechanism. And what will happen then next? This is the main step of this process, that is the repairing. So if the cell will go for the DSB repair, so what will happen in DSB repair? Generally, the cell uses another template strand. As we all know that the most of the organisms are diploid in nature, and they have the two copies of their genetic material. Okay, so this is the chromosome. So we have generally two copies of a particular gene. So if the gene within this DNA get mutated or get damaged, so generally in DSB repair, cell utilizes the another copy, this copy of that particular gene, to repair the damage within this gene. So now at this stage, what will happen if as the Cas9 protein has created the double strand break within this DNA at the point of the gene A, so the cell will utilizes the another copy of the gene on the homologous chromosome and try to repair this gene. Now at this point of time, what will happen? We have added our desired template. Okay, here I have told you that. Okay, this is the desired template. So we have added the desired template within the cell, the sequence template, template which bears a sequence of our requirement. So what will happen at this stage? At some point, the cell will utilize that template sequence to repair this break within this DNA. And just because added template will or the added DNA sequence will act as a template for the repair of this break within this DNA, so it will cause the addition of the nucleotides according to that added template, and in that way we can replace the gene A by the gene B. Okay, the template that we have added will contain the sequence for the gene B, and that sequence or that template will be considered as a template for repairing this break which is created by the Cas9 protein. So because of that, the gene B automatically will be get inserted into the place of the gene A. Okay, so this is how we can replace a particular gene within the genome of an organism with the help of the CRISPR-Cas9 technique. And the double strand break model is very important in this process. Now the second thing is that sometimes generally the cell uses the non-homologous end joining repair mechanism to to repair the double strand break within the DNA caused by the Cas9 proteins. So at that point of time, the non-homologous end joining repair model generally don't uses any type of template strand, but it generally directly joins the two strands of the DNA. For example, like this. For example, like if this is the break. This is the break within the DNA. So what the non-homologous end joining mechanism will do? It will bring these two ends together directly and will cause the joining of the DNA. So it will directly join this broken ends without adding the missing nucleotides. Okay, so because of which certain nucleotides get knocked out, and because of which a particular gene can be knocked out with the help of this model. So this is how we can either we can knock out a particular gene or we can delete a particular gene from the DNA sequence of an organism. So in this way, we can normal gene can be selectively damaged using the CRISPR and altered at high efficiency by experimentally altered gene. So in this way, we can either replace a particular gene with another gene, and we can even knock out or delete a particular gene from the genome of that organism. So this is how the CRISPR-Cas technique is important for the genome editing purpose. Uh, Here you can see for in the embryonic stem cells, like we have added a plasmid which will encode for the CRISPR RNA, Cas9 protein, and our desired template of or the template which will contain the sequence for our desired gene if we want to replace a particular gene. So what will happen that this so what will happen? This protein, this complex, CRISPR complex and Cas9 protein will go and bind to the target site at the site of the target gene, which we want to knock out or replace, and it will cut the DNA at both the ends, and it will cause the double strand break. Now here it will follow the homology directed repair mechanism, in which it will it will replace, or sorry, in which it will repair this break with the help of a template. So as it is, this is the homologous this must be the homologous DNA from the cell itself, but generally in this case the cell is utilizing our added template DNA strand for repairing this double strand break, and in that way this is the sequence which we want to add at this particular site. So as the cell is using this added template as a for repairing this double strand break, so this sequence will automatically get inserted within the break region, and will get a new DNA or the repair DNA with our desired sequence by the homology directed repair mechanism. And if cell will go for the non-homologous end joining process, so it will directly join this broken ends without adding the missing nucleotides, and which will lead to the either deletion or the insertion. It happens sometimes that in certain nucleotides get inserted, and in this way the particular gene can be get deleted from the DNA sequence. So this is much clear from this diagram. So this is how the CRISPR-Cas9 has application for the genome editing purpose. So I hope these things will be clear to you, and if you like the video, please share it with your friends, hit the like button, subscribe to my channel, and thank you so much for watching my video. Thank you guys.